Thank you for your words of encouragement. Very much appreciated. Delighted to be joined with my colleagues, talk about regenerative medicine initiatives, and in our case, the technology that I will be talking about today, cell-based therapies. Usual safe harbor statement. We're a publicly traded company, so I would draw your attention to that statement to the extent that I will be making forward-looking statements today. Is there some way that we can get rid of the two little boxes in the... It will? There we go. So I'm pleased to say that Stem Cells Inc. has programs in all three areas: neurodegenerative disease, both pediatric and adult. In the pediatric arena, lysosomal storage diseases, which were our original excursion into the clinic with our human neural stem cells. Myelination disorders, one of which is PMD, and Steve mentioned that. I will be talking some more about it shortly. And in the adult brain, Alzheimer's disease, which is our latest preclinical research endeavor, and also reference to CIRM. We have received some money from CIRM to help us in our endeavors there. Ophthalmology, age-related macular degeneration is the focus of our targeting here for this same neural stem cell. And spinal cord injury, and in this case our focus, unlike Geron, is in the chronic spinal cord injury patient population. And also, unlike Geron, we're targeting to enroll patients in all three Asia scores A, B, and C, from the most severely impacted to the less severely impacted. The wonderful therapeutic candidate behind all of this is our proprietary human neural stem cell, which is a highly purified population of cells that were discovered by scientists at the company in 1999. These are unmodified adult stem cells. They are not embryonic lineage. And they are highly expandable to commercial scale. They are lineage restricted so that they can only give rise to cells of the central nervous system, and they are non-tumorigenic. In vivo, we see robust engraftment, migration, and differentiation into site-specific cells, in the case of neurons, astrocytes and oligodendrocytes. When I say site-specific, what I mean there is that you will only see neurons in the neuron zip code, astrocytes in the astrocyte zip code, and oligodendrocytes similarly so. And the biological activity of the cells is regulated by the host, and the cells respond to the host cues once transplanted into the host. This platform presents us with a tremendous opportunity for a broad array of clinical applications for a whole host of CNS disorders. A word or two now about the manufacturing, the processing of the cells. As I said, these cells are expandable. They're bankable. This is a cell-based, a product-based cell therapy business model, and we're talking about stem cells in a bottle, literally. We produce a single master cell bank from one donor tissue, from which we derive multiple patient doses. And we go on to make working cell banks and then patient doses. The cells are processed to very exacting CGTP, which means good tissue practice standards, and good manufacturing practice standards. The cells that we have currently for our clinical programs are already banked, and so that's a significant major de-risking of our game plan going forward. And the process, as I mentioned earlier, is scalable for commercial use. I would just like to say that we couldn't have done this without the help of another Alliance member, PCT, with whom we've partnered over the years. And this has been a very productive and very valuable partnership with another Alliance member. And I want to thank Bob Preddy, who I see at the back, who has been personally instrumental in helping us succeed in overcoming some of the technical challenges that we've had in processing these cells. So with regard to the clinical trials, our first trial was in a lysosomal storage disease, NCL, neuronal ceroid lipofuscinosis, or Batten disease, it's also known as. Fatal. We initiated a phase 1 trial in March of 2006. Remember, we discovered the cell in 1996. And we completed that first study in January of 2009. We came out of that study with an extremely favorable safety profile, which of course is job one in this business. And we also had 2 dose cohorts, and we had a significant volume of cells transplanted in the highest cohort, up to 1 billion cells into a pediatric brain. We initiated a phase 1b study in October of 2010, which we subsequently suspended 6 months later, quite frankly, because we could not identify the patient population, we could not identify patients that met the enrollment criteria. And so we made the tough decision to suspend that study until we could figure out whether or not there was a better way to get earlier diagnosis of these children. In the phase 1b study, our objective was to enroll patients who were less severely impacted than the first trial, who had most of their neurons intact at the time of enrollment into the study, because the whole strategy here was a neuroprotection strategy. So we were unable to find patients with this devastating fatal disease who did not have enough neurons present at the time. Then essentially the strategy just would not work. While all of this was going on, we initiated a second study in Pelizaeus-Merzbacher's disease, or PMD. It's a fatal congenital myelination disorder. It's one of the leukodystrophies. We initiated a phase 1 in October of 2009, and we will be announcing and unveiling our results of that study at the end of this quarter. And then spinal cord injury, we initiated that study in March of last year. Thoracic level injury, 3 to 12 months post-injury. And as I mentioned earlier, the patient population in this case, we were able to go from age A's through B's through C's in the same study. So while we shelved the first clinical endeavor in NCL, lysosomal storage disease, we came away from that experience with an awful lot of knowledge. We learned that we could directly transplant large cell doses, and these are allogeneic transplants, of up to 1 billion cells into the human brain, and into the human brain of very fragile patients. And we were able to demonstrate that this approach was feasible and well tolerated. We're also able to show, because we were able to get, unfortunately, brain tissue, because a number of these patients succumbed to the natural course of the disease. We were able to get brain tissue, and we did autopsies. And we were able to show that the cells engraft and migrate deep into the brain structure. We're also able to satisfy ourselves and the agency that there was no evidence of aberrant cell behavior, there was no tumor formation, and that 12 months of immunosuppression was well tolerated by very fragile children. Because of the autopsy data, we also were able to show that these cells survived for extended periods of time post-transplant. And we have data that shows these cells survived for over 2 years. And something that is very, very important for us and very important for the field in general of allogeneic transplant, that the cells endure, at least these cells endured in these patients, post-withdrawal of the immunosuppression regimen, because that was a huge question for the regulators as well as for ourselves, as well as for clinicians and patients. What's going to happen when we cease immunosuppression regimen? So that was certainly a significant observation. Now, myelination disorders, so axons require myelin to transmit nerve impulses. And our cells have been shown preclinically to make new oligodendrocytes and to form donor-derived myelin around host nerve axons. The oligodendrocytes glom on to the nerve axons, and they begin to spin like in jelly roll fashion, series of myelin layers, and there's 18 to 20 of them in a normal axonal formation. Many of the myelination disorders that you know about would include multiple sclerosis and certain forms of cerebral palsy. And of course, the myelin is often damaged in spinal cord injury. And PMD was really our keyhole strategy because PMD is a myelination disorder. It is a genetic defect. The children are born with neurons intact as well as their axons intact, which is very important. But in their case, the oligodendrocytes failed to myelinate, so the axons die and the children die. There is no autoimmune component to this condition, so this was an important consideration for us in terms of being able to protect the cells in this kind of environment. It was a 4-patient phase 1 trial, and it ends next month. And our goal here is, of course, to continue developing the safety profile of the cells in patients. But really what we were looking here to accomplish from the point of view of advancing our knowledge base is whether or not we could show from imaging using MRI techniques evidence of de novo myelin in a human brain that was incapable of developing and deriving its own myelin. And this, of course, would be, if we could succeed in doing so, would be a proof of principle for other leukodystrophies. On spinal cord injury, as a result of a wonderful collaboration with Aileen Anderson and Brian Cummings at UC Irvine, essentially we were able to show that the cells when transplanted into animals who had lost hind limb function, that we were able to restore that function. Not only restore it, but we're also to maintain it. And they also went on to demonstrate that the presence of the cells were critical with regard to the maintenance of the restored function. If you killed off the human cells using a diphtheria toxin, you lost the recovered function and the animals went back to baseline. Very important observation. We initiated a study in Switzerland in 2011. It's open to patients now from Europe and the US and Canada. We will enroll 12 patients who have varying degrees of injuries in the thoracic region, T2 to T11. Apart from, again, safety, our endpoints that we will be looking for will be sensation, motor function, bowel and bladder control. The unique attributes of the trial, its chronic stage, most studies that you see in spinal cord injury in the acute phase. Also, we are able to transition from the most severely impacted to less severely impacted patients. And again, most trials in spinal cord injury get stuck in the Asia A box. We completed our dosing for the Asia A cohort last year, and we're also able to demonstrate to date that we've been able to transplant up to 20 million cells safely into the cord. Our next targets will be in retinal disorders and Alzheimer's disease. Very briefly, retinal disorders are linked to degeneration of photoreceptor cells, progressive loss of these cells in the macula lead to central vision loss. You'll hear some more about that from another colleague who will be up in a moment, Gary. This is the leading cause of visual impairment in folks aged 55 and over, for which I'm a full carrying member. And there is no medical or surgical treatment for dry AMD. Our IND for a phase 1/2 multicenter trial has been filed. Just a word about the preclinical basis for our decision to go forward with this and file the IND. At the ARVO meeting in 2009, we showed that our cells protect the photoreceptors and preserve vision. And we went on to show that these cells also protect the rods and the cone photoreceptors. And that data was presented at the Society for Neuroscience annual meeting, also in 2009. As you can see from this, this is essential. This is what you're looking. This line here is normal visual acuity. This is what happens to the animals with neural stem cell transplanted who have the disease. This is what happened to those animals who only get the media, and this is the natural progression of vision loss in the rat model, which is the Royal College of Surgeons RCS rat model. So this is very compelling data. And last, I'll just finish up with a word about Alzheimer's. Data suggests that the neural stem cells may have utility in Alzheimer's disease. We know from previous work that our cells survive in a plaque-riddled Alzheimer's disease mouse brain. This is unpublished data, it's work we did with George Carlson at the McLaughlin. And publication in PNAS in August of 2009 shows that there is evidence of improved cognitive function in the Alzheimer's disease mouse following the transplantation of mouse neural stem cells into the mouse brain. So this really was an observation that Frank LaFerla made at his lab at UC Irvine. He contacted us and we formed a relationship and a collaboration to bring this work further. We were awarded a CIRM disease team planning grant recently, and we're working with Frank to submit a full disease team application to fund IND-enabling activities in this devastating condition. And our goal is to submit an IND within 4 years. So in summary, Stem Cells Inc. Has successfully transitioned from discovery to preclinical to clinical development, slings and arrows notwithstanding. Our goal now is to replicate in patients the very exciting observations that we've made in very relevant disease models. And I would just like to point out here that in our field of endeavor, the first studies, the phase 1, phase 1/2 studies, are in patients with the disease. These are not in normal, healthy volunteers, as you would see in the small molecule world. So you have an opportunity to evaluate not just the safety profile of the cells, but also to start looking for biological activity, and even in an optimistic mode, to start looking for some kind of clinical effect. So with that word, I'll just finish up and say that we look forward to unveiling the results of our PMD trial in March. And I thank you very much for being here and for your attention. Thank you.